Hello students, this is Baldev sir from Global Education Center. We will be continuing with the concept that is life process. So in the previous video, if you observed, we understood about the various modes of nutrition. Then we focused on the process of photosynthesis performed by the green plants. We understood the steps involved in photosynthesis, and at the end of the video I also mentioned about how various scientists wanted to understand that the requirements or the components of photosynthesis, are they really required for the process of photosynthesis. Now in that regards we understood a process called as test for starch, and we saw that various procedures are involved in testing for starch. Now in order to understand that the components such as the most important ones are sunlight, chlorophyll and carbon dioxide, to understand if these were the requirements of the photosynthesis, there were several experiments that were conducted by various scientists. Now in today's session we will be focusing on those activities that are performed by the scientists to say that sunlight, chlorophyll and carbon dioxide is a requirement for the process of photosynthesis. Also, in today's video we will also be focusing on the nutrition in amoeba. A small introduction to nutrition in animal, and we will be starting off with that nutrition in amoeba. In the next video we will also be focusing on nutrition in human beings. Okay, so let's start. Now, before we perform any of these activities, there is one important or one major step that has to be performed, that is called as destarching. Now what is destarching? You take a particular plant which is going to be used for this particular experiment and keep it in a dark room. Now why do I have to keep it in a dark room? So when I keep it in the dark room, whatever starch that has been present in the plants will be utilized, because in the dark there is no photosynthesis that takes place. Now why do I have to do this? Because at the end of each of these activities, what will we be doing? We will be taking the leaf and testing for starch. Now if I don't destarch the plant will already have a starch, and that will give a wrong result to us. So in order to get a conclusive result, we need to focus the plants on a destarch process, and then we will be performing this activity. So what do we do? First, let's try to understand how the scientists proved that sunlight was a requirement for photosynthesis. So what did the scientists do? They took a plant, they destarched it, kept in the dark room, and then the plant was kept under the sun. But the leaves of those plants a portion of the leaf of those plants were covered by a black paper. So now, if it is covered by a black paper, so when the sun's light falls in this region which is covered by the black paper, there will not be sunlight that will be falling on it. Now, after 4 to 6 hours of this particular plant that is kept under the sunlight, we just pluck this leaf and we will do test for starch. And when they tested for starch, what happened? The region which was covered by the black paper did not turn blue-black, and the rest of the region turned blue-black. Now by this, what can I conclude? The region which was covered by black paper did not receive any sunlight, and because there was no sunlight that was received by it, no starch was being produced. That means the process of photosynthesis did not take place in the region which was covered by black paper. And that is how I can conclusively say that sunlight is a requirement for photosynthesis. Now, second thing that was done by the scientists is chlorophyll. How did they prove that chlorophyll was a requirement for the particular process of photosynthesis? Now this is a very simple activity. Now what did they do? They took a special leaf called as variegated leaf. Now what is a variegated leaf? A variegated leaf is any leaf which lacks amount of chlorophyll in them. For example, crotons. A croton leaf, if you observe, in patches you find chlorophyll. You don't find chlorophyll throughout the region. So now taking an example of the leaf that is already there on the board, the regions here which is represented by the black is a region which do not have chlorophyll. Let's assume the region which is here on the black marking do not have chlorophyll, and the remaining of the region has a chlorophyll. Now again, the procedure remains the same. You destarch it, then keep it under the sunlight for 4 to 6 hours, and then pluck the leaf, test for starch. When they tested for the starch, what happened? the region where chlorophyll was not present did not turn blue-black, whereas the region where chlorophyll was present turned blue-black. So from this, what can we say? That chlorophyll is one of the most important component again required for the process of photosynthesis. Clear? So just concluding this part of the session. So by this test we will get to know that sunlight was a major component required for photosynthesis. How do we do that? Because this part was covered by a black paper, so it did not receive any sunlight, hence it did not turn blue-black. So sunlight is requirement for photosynthesis, and in the case of a variegated leaf, the region where chlorophyll was absent did not turn blue-black, so meaning that chlorophyll is also requirement for the process of photosynthesis. Now, the next activity that was performed was to understand if carbon dioxide is a requirement for photosynthesis. Now, what did they do? They took two potted plants, destarched it, and then they covered it with a polythene bag. They took two potted plants, destarched it and covered it with a polythene bag in order to make sure there is no carbon dioxide, external carbon dioxide entering. In pot A, a particular substance called soda lime was placed, and in pot B, sodium hydrogen carbonate was placed. Now what do this soda lime do? They absorb the carbon dioxide present within this environment. And what do the sodium hydrogen carbonate do? They give out carbon dioxide within this particular environment. So here you need to understand pot B becomes a control for the experiment. So when this particular potted plants were exposed under the sunlight for a brief period of time, let's say 4 to 6 hours, and when the leaf of either of these pots were tested for starch, the leaf of pot B turned blue-black, whereas the leaf of pot A did not turn blue-black. Now what is my conclusion in this? The conclusion in this is that the leaf of the pot A did not have any carbon dioxide, and since it did not have any carbon dioxide, no photosynthesis occurred. Do you remember the third step? That is reduction of carbon dioxide to carbohydrate. Now if there is no carbon dioxide, no carbohydrate, meaning there is no starch that is being produced. Now if there is no starch that is being produced, this particular leaf of pot A will not turn blue-black. Now this is how the scientists concluded that carbon dioxide is a requirement for the purpose of photosynthesis. And with these conclusions, now we have the equation of photosynthesis, that is nothing but 6CO2 plus 12H2O, that gives rise to C6H12O6 plus 6H2O plus 6O2. So thanks to the contributions of the scientists, now we know what are the requirements for photosynthesis. Clear? Now, the next concept that we are going to focus on is nutrition in animals. So we are done with nutrition in plants. We saw how the plants prepare their own food. Now we are all heterotrophs. So when we say nutrition in animals, all the animals are heterotrophs. So we will be focusing on how these animals obtain their nutrition from the plants. Now before going into nutrition in human beings, we will also try to understand how the nutrition in amoeba happens. Now there is a simple diagrammatic representation that is given. Now just to make a note that this particular concept is very important for your board exam point of view. This question, that is nutrition in amoeba with the label diagram, has been asked several times in the board exam. So just focus. So it's a very simple process, that is nutrition in amoeba is a pretty simple process. What happens? An amoeba, it's shapeless. We all know that amoeba do not have a proper shape. That is the better quality of an amoeba. Now also in the previous classes you would have learned that amoeba contains certain finger-like projections called as pseudopodia, which is also called as false feet. Pseudo means false, podia means feet. Now whenever there is a food that is present around an amoeba, the amoeba will try to engulf the food. Now this process we call it as phagocytosis. The process of engulfing of the food by amoeba is called as phagocytosis. How do they engulf it? So basically the pseudopodia, the projections, will throw itself around this food particle and engulf them. So after they engulf it, there is a food vacuole formation that takes place. What happens? There is a food vacuole formation that takes place. Now this food vacuole will release certain enzymes, and thus the food that is taken in, which is within the food vacuole, is degraded and broken down into simpler complex. Now that simpler complex is then utilized by the amoeba, and the undigested food is thrown out by the body surface of the amoeba. Clear? I'll just repeat this part again. What is happening? The amoeba is engulfing the food with the help of pseudopodia. The food comes in contact with the surface of the amoeba, and then formation of food vacuole takes place. The food vacuole contains certain digestive enzymes which will bring down the breakdown of the food, and thus the food is broken down to simpler complex, which is then absorbed by the amoeba. So this is how the nutrition in lower organism, to be more specific, in amoeba takes place. So with this we conclude with this session. In the next session we will be focusing on nutrition in animals, to be specific nutrition in human beings. We will be seeing the various process that is involved in the breakdown of the food that we consume in various parts of the digestive system. So have a great day, great week, until next time.